Good morning and welcome. I'm Bonnie Raindrop, Project Director for Pesticides and the Chesapeake Bay Watershed, and I'm very happy to welcome you to this program today. Sorry. Very happy to welcome you to our program today, and we're so glad that you could join us. This project conference has built a really unique, diverse community, and many of you attend year after year and are members of our project working groups. Some of you may be first-timers, and we're glad to have you joining us today. All of us are concerned in some way with the impacts of pesticides on the Bay watershed, and you all are experts in your own fields. The Watershed Project is unique because it brings diverse stakeholders like yourself together to learn and collaborate to advance our understanding of the role pesticides play in the watershed and how we can work together more effectively to address pesticide impacts. So thank you for taking the time to be here today. Next, before we move on, I want to do a little bit of housekeeping. Please open your chat window at the bottom of your Zoom screen and use the little arrow to select Chat to Everyone. You can also change that chat toggle to chat to the panelists only. And now please introduce yourself and share something about who you are. I think we all miss the networking we used to do when this was an in-person conference, but we'll do the best we can with chat. So throughout the conference, please use chat to share your thoughts today as we hear from our presenters. I'll send out the chat transcript after the last session, and you'll also get a roster of all attendees with their contact information to help you with the networking that we like to do. And when you use this chat, the networking opportunity is really foremost. So be generous with what you share about yourself and your interests. You may even meet a new colleague in this process. We'll also use the Q&A module to pose questions to our presenters. So click on that Q&A icon at the bottom of your screen and type questions in as we go. Remember, if the question is for a presenter, you need to use the Q&A tool. If you put these questions in chat, presenters may not see them. So if we don't get to your question live, presenters will stay on after the live Q&A and respond to those questions in the chat tool, and then we'll send a transcript along with that next week. So around 11 o'clock, we're going to pause for 5 minutes so you can take a short break. And when you come back, we'll have a chat question on screen for you to answer. Next. Now I'll turn things over to our conference moderator, Dr. Greg Allen, who's been our moderator since the Watershed Project inception 17 years ago. Greg also has led the Research and Data Gaps Working Group for just as long, and now he shares that role with Dr. Vicki Blazer. But if you don't know Greg, he's an environmental scientist at the U.S. Environmental Protection Agency, the Chesapeake Bay Program Office, and is approaching a 30-year anniversary at EPA. Greg is chair of the Chesapeake Bay Program's Toxic Contaminants Work Group and coordinator of the Partnering, Leadership, and Management Team. For nearly a decade before joining EPA, Greg was in the field and at the analytical chemistry bench, analyzing the occurrence of organic contaminants. Greg counts among his most meaningful recognitions a White House Closing the Circle Award, an EPA Gold Medal, and a handwritten thank you note from the Puget Sound Partnership for helping with their PCB reduction strategy. So welcome, Greg.
Thank you so much, Bonnie. And good morning, everyone. Welcome to part 2 of this year's conference. If you were with us in the first session, welcome back. And if you're joining us for the first time this year, welcome. I think you're going to really enjoy today's conference, and thank you to Bonnie and team for once again putting together a wonderful slate of presenters and discussion topics for us. I think you're going to really enjoy our time together today. And to get us started, we are going to talk about some of the things that have sustained this project over time. One of them is that we have work groups that produce valuable work products, and we're going to hear about that work today. And the other thing is these common shared values that we use in coming together to work on this issue. And I'm going to pause for just a moment so that you can read this list of our shared values. So using these values has ensured that we have constructive, productive conversations. And that's been true now for 17 years. And we don't expect that that's going to change anytime soon. And we are looking forward to the 18th annual conference. So let's be sure to keep these values in mind. And the next thing we're going to do here, just to get everybody talking to one another, is we're going to ask you a chat question. So here's our first chat question today. What is one goal that you could set for the new year to contribute to a healthier Chesapeake Bay and watershed? We are in the holiday season. This is a time of year when we set goals and resolutions for the new year. We just want everyone to think about one thing they could do to make for a healthier watershed and a healthier environment. If you're joining us from outside the watershed, think about your own local area. I just want you to share one thing that you could do in the new year to contribute to a healthier bay and watershed. And I'm going to pause again for about a minute and a half or so to give you a chance to answer the question, and follow along with your colleagues in the chat. Maybe you'll be inspired by someone else's goal. I'm going to pause for about a minute and a half while you answer this question. Well, those are some absolutely wonderful goals that are appearing in chat. Thank you everyone for contributing to that. Wow, there's so much inspirational in that list. I saw things having to do with educating young people, our students, on environmental issues, things related to new policy, adjusting our own diets, maybe to be a bit more on the organic side, interest in topics like plastics and PFAS. We'll be hearing about PFAS today. Native plants, oyster restoration, really like the one that said 1 acre at a time. 1 acre at a time, because when we face really big challenges, the way to make progress is breaking it down into small wins, doing it one step at a time. Wow. Okay, great. Thank you all for that. I hope you'll maintain your commitment to those goals, and maybe you are inspired by your colleagues. Continue to put your ideas in chat and follow that chat and hopefully be inspired by that. Okay, well, let's take a look at the program for today. Wonderful set of presentations we have. So in our second conference session today, we're going to continue with the theme of the interaction of pesticides and the soil and human microbiomes. And we'll start today with information on restoring a healthy soil microbiome. Orion Valcourt of Mid-Atlantic Microbials will share some examples of soil restoration on Maryland's Eastern Shore using practices that we learned about in our first session and hearing from Dr. Elaine Ingham. So we'll see some examples of application of those techniques. Next, we'll hear from Catherine Dalton, who will focus on the human microbiome and how interactions with pesticides may be affecting our own health. The project includes our ongoing work throughout the year from participants who collaborate in our 3 project working groups. We'll get a report from them today about what they've been working on and how they've been working with your ideas that we captured 2 conferences ago. If you've been with us for a couple of years here, you might remember 2 years ago, we did a really large scale Jamboard brainstorming activity, and we are still benefiting from your input and still using that input to help ensure that our work groups are working on high value projects. So we'll hear about how our work groups are continuing to mine that information for their work plans. And then we'll close today with a session on PFAS. And this is an update from Nathan Donnelly, who built on what we learned last year. We had PFAS and pesticides as a major theme, but we're going to get a wonderful update this year from Nathan. All right, well, that is our program for today. So let's jump right in here. We're going to start with presentation from Orion Valcourt. Orion has been serving as the managing partner and operations manager of Mid-Atlantic Microbials since 2018, collaborating closely with his wife, Jewel Valcourt. Their shared passion lies in advancing the understandings of the intricate science behind soil microbiomes and utilizing this knowledge to accelerate the process of soil restoration. Fantastic. Okay, welcome, Ryan.
Good morning, everyone. I'm very, very excited to be here. Thank you for the wonderful introduction. So here at Mid-Atlantic Microbials, we, as you learned in the first session from Dr. Elaine Ingham, we are a soil microbiology amendment company that does not only soil amendment, compost, but we also do liquids. So in a nutshell, I want to share with you all some of the things that we have done here in the Mid-Atlantic region, mostly all here on the eastern shore of Maryland. Slide. So I'm going to do a few comparisons. So right here we have a row crop farmer, not from our area, but a project that we had done quite a while ago. And this was done with our dry soil amendment. And as you can see in this photograph, the standard conventional practices are on the left. That's a 65 bushel an acre standard for this particular farmer. The soil amendment that we used, and this was a one-time application, had a significant increase in yield up to 97 bushels an acre. So you could imagine that the farmer was very, very happy with that. And when you look at the comparison of the plants and the plant material, you'll see that the soil amendment only treated side there is much more dense in its foliage and of course the food production. And when you're growing in a healthy soil system, as you learned in session 1 from Dr. Elaine, who is one of our science advisors since the very beginning of this company, you get a much healthier plant that is resistant and needs no pesticides or herbicides. In addition to that, the nutrient density of the food being produced here, in this case it's soybeans, is dramatically increased. So when you get to the situation of food insecurity, which we're all very aware of, having plants that are growing in this type of environment, you're going to remain a much higher nutrient density, and the microbiome of our body is a mirror of the microbiome of the soil. So you're going to absorb a lot more of the nutrients and you have to eat a lot less food. So I just wanted to make that point right here at the very beginning. Next slide, please. So here is our St. Paul's church restoration, and this was a stream bed area. Slide, please. Now they used spent mushroom soil on this site, and you can see right here that spent mushroom soil has been put down. Next slide. So this is 2 weeks after. You can see how the grass is taking off. Next slide. And this is a picture of the control area. So you see where there's no germination happening is where they just had the spent mushroom soil and seeded it. The area where you see all the grasses growing is where we treated it. So on this site, we used not only our dry application, which is our soil amendment, but we also used liquid extract. So doing a liquid extract requires us to strip the biology into a liquid form and then deploy that. Our recommendation to farmers or restoration projects is 16 to 20 gallons an acre. Next slide. So this is more of a close-up. So you see the before and the after, and in 2 weeks you can see how dense the germination was, and that is coming into a grass maturity. Next slide. This is Tuckahoe State Park. This was an agricultural site that was being returned back to nature. Next slide. So it's a little difficult to see the control in this picture where there was no treatment. And the treated area has a much denser foliage and germination rate. Next slide. So this is a close-up of the control, and you can see that there's some indigenous weed, some bare areas. And when you look at the treated side, you can see how much denser the germination is, and this is weed-free. Next slide. So this is a project that we did at Camp Pecomath, and this is the stabilization of a eroding beachfront. Next slide. So we came into this project after they had already put the grass plugs down. We came in after it was done and then had to amend each one of those areas. So you can see that beginning of that on the slide to the right. The December 20th slide. Next slide. So here you can see 6 days after application on December 26th that we're getting germination in 6 days. And this was in December, so not ideal conditions for germination. And then the companion picture here, 10 days after application, you can see that it is absolutely starting to take hold. So this is 5 months after, and you can see that the treated area is now really starting to establish, and realizing this was winter, so things are a little bit slower to establish. And then the south end here, where we did no treatment, our control area, you can see how sparse that is, and it's just barely getting going. So this is Caroline County Parks and Recreation. This is a wildflower restoration, is the part that we did here. This is a private-public partnership. We worked with all the engineers to do this berm. Next slide. So on October 28th, you can see this was right after seeding, and they had put down the hay there. They had used on this site topsoil that they had stripped, and the topsoil, after we looked at it under the microscope, was not very robust topsoil. So you can see on 18 days later, after we did our liquid application, we were discharging our hoses, and you can see that it left like a hieroglyphic behind. Next slide. This was before our liquid application, and our soil amendment and compost was mixed in to this berm site. So this is 18 days after, and these are areas where we applied. You can see that it's still a bit rutted up. They used some, heavy equipment to create these, and so there's some compaction areas. So anywhere where you don't see it germinating really well, it was a bit compacted from the tracks of their equipment. So this is another Caroline County site that we did, and this is an example of athletic fields. So what we did here was we put down our dry amendment, and then we went over it with a liquid treatment. And you can see that with one of our partners, Todd Higgs, is pulling the tank while we were spraying that. So this was a very unique situation. They were thinking of spending over $100,000 to put down new sod. And I told them, I said, look, let's go ahead and see if you aerate the field. We put down our dry amendment, and then we come do some liquid treatments. Let's see if we can bring these fields back to life. So this is what we saw under the microscope. So the control sample is pretty indicative to the Eastern Shore. It is got a lot of sand, silica there in the sample. It's bacterial dominated. You don't see any fungal hyphae. It's in very poor condition. The two treatment side, that's after our second liquid treatment, after our dry went down. And you can see this is much more robust. The system is coming alive. There's fungal hyphae in here now. You've got the majority of all the good guys you want in the bacterial and fungal. We also found the nematodes, the micro and macro orthopods, all on this site after our second treatment. And this was only 3 months, 3 months in. Next slide. Here we have Mason's Farm. That's Bill Mason started this as an organic farm, and Steve, his son-in-law, who had brought us in several years ago, and we have treated around 250 acres there in different test areas over these last years. We've done the majority of it has been done with our liquids only. One real interesting thing here at Mason Farms is that we were able to deploy our liquids through their overhead pivot. And that was that's a 100-acre pivot that they had. So we can deploy in large scale on large farms to get the biology down in a drench. And we also do foliar applications in the event that there is a pest infestation that is recognized. We can create a foliar spray also. Next slide. So we've done the dry amendments, 2 liquid amendments, and we also have quite a bit of microscopy work that we've done here to track how this is going. Next slide. So here we have the before and after microscope imaging. The control sample you can see is pretty similar to the one we looked at earlier. It's pretty much full of silica. Yeah, it's sandy soil, bacterial dominated. And then the 3 treatment sample is where we're really building soil structure. This is now about 18 months in, and we have fungal hyphae. We've got the nematodes, macro orthopods. It's coming close to being a balanced soil system. Really beautiful work over there at this organic farm. So questions from anyone? Happy to answer any questions. Thank you.
Thank you, Orion. Wow, really wonderful. The pictures that speak 1,000 words. So thank you so much for that. So we do have a couple of questions for you, but before we get to the questions, we're happy to let you know we have a surprise guest today. And you might recall last conference when we had Steve Kraszewski from Mason's Farm, which is Maryland's largest organic grain operation. And Steve gave us a presentation on his farm's adoption of organic regenerative practices, and it was really wonderful. So great to see this partnership and the team between Orion and Steve, but great to have Steve back this year. And Steve, we're just going to give you a couple minutes to give us a quick update about things happening on your farm.
Very good, Greg. Thank you for that introduction. And yeah, the relationship between Mid-Atlantic Microbials and, of course, Orion, kind of started with that partnership, as you mentioned, based on the same, I guess, the same philosophy, which is kind of improving from the ground up the soil, or as I think Elaine and Adam's presentation from the beginning of the month, the first session, converting dirt into soil, or using that similar analogy. And we were just needing a change here on the farm, and we were able to, yes, get in contact with Orion and the company. And what made it very helpful for us, especially on the farm at any time, they can do those assays or microscopies in real time. Come on the farm, grab the sample, they take it back to the lab, to their lab, analyze it, and put together a custom tea or drench with the proper microbial, the organisms, and have something available to you in the next several days, or possibly even the next day. That was a very helpful thing for us. And so, yeah, analysis and the solution in real time, especially when we talk about soil health practices and regenerative farming. That's a big deal for us and anybody else who is using this kind of system. But what the main thing I wanted to say about Mid-Atlantic Microbials, they're able to put those concepts and the science that you learned from Elaine's presentation last week, and put those into practice and implement it on a large field scale. For example, as Orion mentioned in his presentation, using the center pivot, which is actually a 200-acre circular pivot irrigation system, and as simple as just hooking in some hoses and running it through as a drench on the acres that we want to. So yeah, that's an easy, quick way to scale up. And we also, from you noticed from last year, we can also apply it with a sprayer. And just like any of the other equipment that covers lots of area in very quick fashion, can be done quickly and easily. Liquids, easy to administer, and top dress your field and so forth. And so, but we've really seen some positive results from with these theories and claims. They can really back them up. Our strip till corn experiment went very well this year with the tea drench, and saw some better yield gains than any of the other types of products we put out in the row. So we're very happy to see that. And not only the end of the year with the yield, when the combine goes through and you get those numbers, but during the season, we could see improved drought tolerance when we had dry spells. Even though these fields are under irrigation, we noticed that we could let the irrigation go even longer if we chose to, without seeing the normal stress that we could from fields right next to it that didn't have any of the similar treatment that we're not currently experimenting with these teas and these microbial inoculants. So those were some very positive results that we were able to see. And yeah, it's all about building that robust, resilient, as I call it, a soil reservoir. That's what these products can do. It's just a given time and a plan. We just have to make sure that our management practices in the field match what we want to see. So reduced tillage has been our main goal, and with Orion's help and Mid-Atlantic Microbials and their product, and that's we're on our way here at Mason's Heritage.
Fantastic, Steve. Thank you. So great to hear about your continued progress there and the scale at which you're able to demonstrate and use these practices. Really fascinating. Okay, well, we've got time for a couple of questions here. Probably back to Orion, just folks asking, could you say a little bit more about the formulation of both the dry and liquid application? What exactly is in those products?
Well, our dry amendment is derived from the massive excess of chicken litter. It doesn't need to, so we set up on chicken farms, and we use that as our basically nitrogen base. We mix that in our proprietary blend that took us over 7 years to create, with a hardwood chip that's coming from all the landscapers and so on. So yeah, these are inputs that are accessible, they're easy to get. We also have some green materials that wind up going in there, but because we're able to work with the microbes, we put that into a composting system that I don't want to go into because that'd take a little bit longer. And so this is all indigenously based. All the chickens are here on the Eastern Shore. The plant material that's coming in to create the compost is all indigenous. And I saw one question that said, do we do that? Yes, we mine the indigenous biology from the forest around the farm, like over Mason's farm. I took a couple buckets, 5-gallon buckets, took a walk in their woods, right? And we wound up creating some of our biology from exactly what is right in their field area. So does that answer the question?
Yeah, it sure does. Now, the wet formulation, is it just the same as dry, but in a slurry, or what's the difference between dry and wet?
Okay, so what we do is we take our compost and or amendment, because we do amend our compost to create a really, really powerful immediate reaction. And so we strip that and in an aerated tank. So there's air bubbling up. And so a drenches takes about 2 or 3 hours to extract all the biology. And then that can sit for a few hours or whatever. It's not in a rush to get it right out the door, but it has to go down within 24 hours. So we make it on farm, and or like at Steve's farm, we're really close. So we make it at our, in our brewers, and then just run it over there in the morning, and, he puts it out. So, yeah, very simple. We make these in batches of anywhere from 250 gallons to 1,000 gallons. As I said in my presentation, 16 to 20 gallons an acre is the application rate. And that is a drench. So a foliar application, it takes 16 to 24 hours. And then what we're doing is we're growing the biology species that we want in the tank. And then that's going to stick to the plant surface. So no need for pesticides or herbicides if you're doing a foliar, if you see something that you need to use a foliar for. We've not done that at Mason Farms yet.
Okay. All right. Wow. Thank you. That helps us understand the formulation and what we've got in there. So yeah, unfortunately, there are many, many questions coming in. So much we'd like to ask you about. I'm going to just grab one here from the list as we conclude this discussion, and it's about cost. Where are we at cost? I could expect that we're at a smaller scale at this point, so maybe we haven't gotten all the benefits of scale yet, but do farmers have a cost-effective alternative here with managing their land yet, or are we still in a scale up to get that cost down?
Well, I'll make a quick comment on that. Our cost is $1.25 a gallon. So when you're putting down 20 gallons, comparatively to, and Steve can answer this question, to what a nitrogen product or any other organic fertilizer that they use there. So all our studies show that we are significantly lower cost. So Steve, any comment on that?
Yes, those numbers are right, Orion. So anywhere between I think $30 and $40 an acre, depending on, yes, the rate we put down, which is about 20 gallons per acre. But in terms of what you get for your application, you're talking about, if you're putting down, say, poultry litter, that's one of our sources of nitrogen and other fertility. We put down, for at least for our corn, you're looking at a minimum of $100 per acre for every acre for your corn ground, just your corn ground. And so, even several applications of the product from Mid-Atlantic Microbials and what you're getting out of it, as long as you are managing your field the way, like I said, that you're trying to achieve your goals, as long as you're keeping your tillage down, that's probably the most important thing. As long as you're letting those trenches do their work, letting the soil be at rest, and watch your compaction and many, many other things along the way, it's seeming to bear fruit, and it's well worth the investment.
Wow, that sounds really encouraging. Okay, well, thank you.
I do want to make one comment real quickly, is that this hasn't been brought up that I know of, but the carbon sequestration level of a healthy soil environment is off the charts comparatively. So if, when we're looking at global impact, if we can restore farmland, like we're doing, with Steve and others, and or recreational spaces, urban space, it doesn't matter. You're going to get a tremendous amount of carbon stored, and it's going to stay there for thousands of years. I mean, as long as it's an active microbial soil environment. So, yeah.
Okay.
Just wanted to get that last one in there.
Well, that's a big one. That is a big one if that potential exists. Wow. Okay, great. Well, thanks again, Orion and Steve. There are some additional questions in Q&A. If you can take a look at those and provide some answers, we would appreciate that. But we're going to move on now. We're going to move to our next presentation. I'd like to introduce Catherine Dalton. Dr. Catherine Dalton is an epidemiologist, an environmental microbiologist, and veterinarian who studies environmental factors that impact health through the role of the microbiome. Her research seeks to promote One Health, the shared health of humans, animals, and the environment. She's currently a postdoctoral research fellow at the National Institute of Environmental Health Sciences at NIH. And so we are just so pleased to welcome Catherine. Catherine?
Hello, everyone, and greetings from North Carolina. So far in this conference, we've heard some really interesting talks about the effects of pesticides on the microbiome in soil and in the environment, and how that impacts ecosystem health. But today, I'd like to shift gears a little bit and discuss more about the role of pesticides and chemicals in general on the human microbiome and how that impacts public health. Next. Before I get started, I have a standard disclaimer that views are my own, not NIH. Next. Within this talk, I'll start by reviewing key concepts of the human microbiome and its role in health, and then delve into the role that pesticides play in this relationship. And I'd like to end with discussing future directions that I believe are needed in this field to promote public health. So we've heard talks describing the microbiome already as part of this symposium, and I'm sure many of you are already somewhat familiar with the concept of the human microbiome. It's frequently featured in the media, and there are a ton of products coming out now that claim to be good for your microbiome. But what does that actually mean? What is a healthy human microbiome? Next. Well, in general, a healthy microbiome is a diverse and balanced community of microorganisms that function as a holistic system. A diverse microbiome is both rich, or having many different kinds of species, and even, or having those species be relatively equal in abundance. This is important because each microbe will have an important function that they play in the body. And so if there are disturbances to the community, you have more microbes to fill those important roles. With regards to human health, we know that our microbiome has critical roles in immune regulation, metabolism, neurological and hormonal signaling, and overall homeostasis. Next. As such, our microbiome plays a critical role in our overall health, and disturbances to the normal balance of our microbial communities can lead to various disorders. These include gastrointestinal and neurological disorders, atopic and hypersensitivity conditions, liver, renal, and skin conditions, infectious disease risk, and many, many others, beyond what is shown in this figure. There are actually very few disease conditions that in at least some way is not related to the role of the microbiome and their microbial metabolites. And we are continually learning about new associations of our microbiome to our health. Next. Now, I want to clarify that it's not just the microbes in our gut that are related to health. While our gut microbiome is obviously a very important site, and it's probably the community most extensively studied, mostly because there are more microbes colonizing the gut compared to other sites in our body. However, any site that has even a little contact with the external environment will have microorganisms present, which will have the same key functions as our gut. This includes the respiratory tract, the upper GI tract, such as the oral community, our skin, the vaginal microbiome, plus a handful of other ones. You can see in the figure on the right that each community at a different body site, shown on the bottom axis, will have a different composition and balance of microbes. Some are listed on the side axis, where a larger circle means they are more prominent in that location. Next. Now, I want to take this one step further. It's not only the microbiome in our body that is linked to human health. As we've learned in previous talks, we are surrounded by microbes in our environment. And they are not only in the natural environment, in soil and plants, but also in the built environment, such as our homes where we spend the most time. And these environmental microbiota work to influence our health through interactions and potentially changing our own microbiome, as well as directly impacting health processes, such as in the case of antigenic stimulation and infection. Next. Adding to this overall complexity, we know that there are many preceding factors and drivers of our microbial communities, both in our bodies and in our environment. These can include lifestyle factors, such as diet, medication usage, and other people we interact with, our genetics, our early life experiences, such as mode of delivery, and our physical environment, both green spaces and adverse pollutants, including chemicals and pesticides. And so this leads us to the next section of my talk, where we'll discuss the interactions between these chemical exposures and our microbiome. Next. One factor that has been less studied in reference to human health is the complex interactions between chemical exposures and our microbiome. In fact, the National Academies put forth a report back in 2018 that emphasized this field as a key future direction needed to advance population health. Next. There is a very interesting bidirectional relationship between chemicals and microbes that I will break down. First, we are exposed to chemicals which can potentially change our microbiome. We know that various chemicals have antimicrobial properties, where they can kill select microbes. Conversely, other chemicals can promote the growth of certain microorganisms. Overall, this creates an unbalanced community, or dysbiosis. As I alluded to earlier, this will likely have consequences to the important roles that our microbiome fills in health. We have seen this in both controlled lab animal models and epidemiological studies in people from the effects of BPA, phthalates, persistent organic pollutants, endocrine disrupting chemicals like triclosan, and heavy metals on the composition of the microbiome, although most focus on the gut. Next. On the other hand, when we are exposed to these chemicals, they are metabolized by our bodies, and it's typically the metabolites that cause the toxic effects. But our microbiome plays a critical role in metabolism and can alter the absorption or metabolism of these chemicals, whether it be in our gut or oral communities for ingested chemicals, or our nasal and respiratory microbiome for inhaled chemicals. And this can potentially change the toxic effects in our body, making them more or less toxic depending on our microbial composition. So overall, our microbiome can mediate the toxic effects of chemicals. There are fewer studies in this area, most are from laboratory models, which show decreased chemical metabolites and reduced bioavailability related to the presence of certain microorganisms and microbial functional byproducts. Next. Now, with specific regards to pesticides, as that's the focus of this talk, there is still a lot we don't fully understand within this model. Next. First, we know that the microbiome itself is closely tied to our health, and changes to the microbiome alters health outcomes, as we've established in the beginning of this talk. Next, it's been pretty well established that pesticide exposure leads to adverse health outcomes in both agricultural workers and the general public. I could have a whole presentation on various health outcomes in people related to pesticide exposure. I'll review some highlights from a particular study called the Agricultural Health Study, which is a prospective cohort of over 50,000 licensed pesticide applicators and their spouses in North Carolina and Iowa that have been followed for over 30 years. To date, the numerous studies coming out of this cohort have demonstrated that pesticide exposures have been linked to a whole suite of adverse health outcomes, some of which are listed here. You can see there's a big range of health effects, from cardiometabolic disorders, autoimmune conditions, degenerative disorders, respiratory conditions, birth conditions, neurological and psychiatric disorders, a range of different types of cancers, and overall increased mortality. Now, that's not to say that every pesticide causes every health effect shown here, but at least one pesticide has been linked to at least one of these outcomes, and most pesticides are linked to multiple health outcomes. Next. Lastly, and what I want to focus on, is that we also know that some pesticides have been shown to alter the microbial communities in our bodies and in our surrounding environment. However, this has been less well established than the previous 2 points, and is an area of future research growth. In controlled experiments in lab animal models, researchers have found altered gut microbiota when exposed to glyphosate and chlorpyrifos. These are the 2 studies most frequently cited. However, there have been other lab-based studies showing similar trends. Next. In human epidemiological studies, a recent report this year in the Dutch Microbiome Project of Lifetime Studies showed altered gut microbiota in occupational workers exposed to insecticide and fungicides, specifically lowered microbial diversity and increases in microbes associated with disease conditions. A birth cohort out of Brazil found changes in both maternal and infant gut microbiota related to maternal exposure of DDT and pyrethroids. Another set of studies in Washington showed changes to the oral microbiome of farm workers and their children related to blood levels of organophosphate pesticides. Next. Lastly, my colleagues and I have explored the influence of pesticide exposure on the microbial communities inside workers' homes. We found that exposure to the pesticide glyphosate in the last year, as well as past exposure to banned organochlorine pesticides such as lindane, were associated with decreased microbial diversity levels in homes. And we found specific microbes that were different in their abundance by pesticide use, specifically current use of 2,4-D and atrazine, and past use of heptachlor and lindane. The decreased microbes tended to be common soil-based ones, such as Haloterigena, while the ones that were increased could possibly be pathogenic to people, such as Fusarium and Toxoplasma. Next. However, while work in this topic is still growing, there has been no research to date that has incorporated all of these aspects and the links between them to truly understand the mechanisms of how pesticide exposures cause health effects through the pathway of the microbiome. Next. And as I discussed earlier, there is a dynamic bidirectional relationship between our microbial communities and chemicals, including pesticides, that should be considered in the context of human and also ecosystem health. No study has examined if the microbiome composition alters the toxic effects we see from pesticide exposure. Next. Additionally, we know that different pesticides have different chemical properties and cause different physiological changes in our bodies. And the combination of various pesticides may have differing effects, which may result in unique impacts on the microbiome and on our health. Next. Lastly, as we've heard from speakers in our previous sessions, and will hear from following ones, that pesticides can contain other harmful chemicals such as PFAS. Plus, we are continually exposed to these harmful chemicals like PFAS in our environment. And so the mixture of multiple chemicals and pesticides together and how they act synergistically or antagonistically in our bodies, and then how this interaction relates to changes in our microbiome, is important to assess. And so I've provided the rationale and background, as well as the current knowledge gap in relation to pesticides and our microbiome. But why is this so important to understand and address these knowledge gaps? Next. Well, I'm a researcher, so I will always argue for understanding a scientific phenomena for its own sake. But in this case, there is a direct practical application to truly defining parts of this system and how they are related. Understanding the effects of pesticides on the microbiome and vice versa can provide valuable insights into the mechanisms that underlie the impact of pesticide exposure on human health. Pesticides, like other chemicals, can be ubiquitous in the environment with widespread contact, and so can be challenging to reduce their use and exposure. If we learned anything from history, it's that even if industry does come up with alternatives to adverse products, sometimes these alternatives can also have detrimental effects that we are unaware of. So what can we do to reduce the adverse health outcomes from these widespread pesticide and other chemical exposures? Next. Well, that's where I believe the future of the field of microbiome science can come into play and be a valuable asset in potentially solving this conundrum. Buffering the microbes in our bodies and in our environment, such as our homes, can be a novel mechanism to ameliorate the toxic effects from pesticides. By creating a rich, diverse microbial community within our gut, oral, nasal, skin, reproductive sites, etc., and the microbes in our environment, our microbiome will be more resilient to changes related to pesticide exposure, and we can possibly alter metabolism of these chemicals and reduce their harmful effects. Now, this is a very novel area and approach, and there really has been no studies yet to evaluate this. Next. However, we can borrow lessons learned from the area of drug trials, where there has been a little bit of research in this area. Researchers have augmented germ-free mice with a robust microbiome and found that they responded better to the anesthetic phenobarbital and were less tolerant to opioids. I believe this data hints at the possibility of the microbiome as a future intervention point to help reduce the adverse exposure to chemicals like pesticides. But I acknowledge that this is a very future goalpost, hence why I titled this slide The Moonshot. Next. So I hope that in this talk, I've convinced you to consider the role of the microbiome when assessing the human health impacts from adverse pesticide exposure. The microbial communities, both in our bodies and our surrounding environment, perform key roles in physiology, and are such intricate parts of our overall health. Hence, they can serve as valuable mediators and biomarkers to assess chemical and pesticide effects. I've proposed in this talk a sort of roadmap to investigate the possibility of the microbiome as a future intervention target to advance public health as it relates to the adverse effects that we know exist from chemicals and specifically pesticides. While there is still a lot of work to be done in this area, I believe this can be a novel and exciting field in order to protect and promote public health. Next. And so with that, I would like to thank you all for your attention, and I would be happy to take questions at this time.
All right, wonderful. Thank you so much, Catherine. You really make a strong case for the importance of a healthy human microbiome and potential interactions with pesticides. So fascinating information. We have just a couple minutes for a couple questions. So this first question for you now, we're bringing it back to the farm community, which we've been talking a lot about in this year's conference, and just wondering, what is the strength of science to show that there's a difference between the health of farm families and farm workers using standard versus some of these more organic based soil health based practices? Are there studies that show the difference between standard and more progressive practices on the farm and farm family health?
Yeah, that's a great question, Greg. And there absolutely has been some research in terms of different health outcomes related to whether farmers use more traditional large-scale practices versus more organic practices. And specifically, some of these outcomes do relate to the microbiological outcomes, such as the prevalence of drug-resistant bacteria, not just in the farm environment, but in the workers themselves. But as far as I'm aware, there has been no studies that have explored the effect specifically on the microbiome and how different practices and husbandry management styles impact the microbiome of not only workers, but also the greater community, the workers' families as well. But I do think that could be a very interesting avenue to further explore, because knowing that these other health outcomes we are seeing changes in, I do anticipate that we'll see changes in the microbiome as well. So that's a great question.
Okay. Future directions for research there. Okay. So one more question here. If you were to give your top couple of tips and suggestions for how we can all maintain a healthy human microbiome, what are a couple of your top suggestions?
Oh, that's a tough one. I would say we know that in general, having a diverse microbiome is beneficial. And so really just getting out there and having different experiences and exposures can be beneficial. And so particularly in early life, we know that kids that are raised on farms and these more vibrant natural environments do grow to have more robust microbiomes and are, associated with decreased diseases related to the microbiome. And so that, I think, will really help individuals out in their future. I know there's a lot on probiotics, things like that. It's a very unregulated field. So I would be hesitant to, widespread recommend probiotics. However, I think some of them are actually very beneficial. And so, yeah, I definitely recommend, particularly dietary probiotics. Whether these other topical ones, I am less certain of, but absolutely, I think that could definitely help as well too.
Okay, Great. Well, that's inspirational to us, and we can all strive for homeostasis, right? That's what we're looking for in a healthy microbiome. So thanks for those tips and all that information, future direction. So glad that we were able to pull this factor in. It's really important. So thank you so very much for joining us today and for your presentation. Once again, there are some questions in chat that we're not able to get to. So if you can visit those and provide some answers, we would appreciate that.
Will do. Thanks so much, Greg, for having me here. I really appreciate it.
Yeah, thank you, Catherine. Okay, well, we are up on our break. So we're going to pause for 5 minutes right now and take a break. We will see you back at 11:13 in 5 minutes. Welcome back, everybody. We're going to continue with our program here. We're going to start next with your second chat question. And this may be one that we need to answer over a few minutes. So again, we're going to leave this open so that you can contribute as you have ideas. And what we're asking here is about the application of the knowledge and information we've been sharing during the conference. How can we apply this knowledge of healthy soils and our body's microbiome to the way that we live and run our businesses? How can we apply this knowledge? Now, I will again pause for about a minute and a half, give you a chance to write something in. And again, we'll leave it open. You can continue to contribute to it throughout the rest of our time today. So let's take a shot at this question. How can we apply this information? I'm going to pause for about a minute and a half. Thanks everyone who has shared in chat. Another fabulous list. I see in there that we can apply this knowledge in our organizations, as consultants, as business owners, in our families, and for those that we care about. See, applying it in our own gardens, supporting local growers or using more environmentally friendly practices, and overall just being more mindful. Yeah, so thank you, Bonnie. Being more mindful about our own microbiomes and how important it is in regulating our health and being sure that we're taking care of it. Well, those are a lot of wonderful applications in chat. Please keep adding them because I know I can say I am inspired by those ideas and those commitments. So keep building them in there, and thank you very much for those who contributed. Okay, so let's move on to our next element here. In this project, we have a 17-year history of bringing diverse stakeholders together to share knowledge and information and collaborate and understand and mitigate the impacts of pesticides through our 3 working groups. These working groups are a collection of people that often come together from our annual conference. In other words, we tend to recruit and encourage participation and find people that we need to pull into these work groups. So you're going to hear several times in the next few minutes a call to action, an invite for you to join any of these groups if you're intrigued by the work that you're going to hear about. So we have 3 groups. There's the Research and Data Gaps Work Group, comprised mostly of state and federal agency scientists, also academic researchers and others. We've got the Healthy Alternatives Work Group. They're advocates and educators. They focus on public education campaigns around solutions and healthy alternatives. We've also got the Sustainable Agriculture Work Group. That's comprised of farmers and beekeepers, agriculture and land care professionals, business consumers of ag products, a diverse group working on materials and products that will help promote sustainable agriculture. So what we'll do now is hear from each of the groups, just a real brief update on their progress over the last year, so since our last conference, some ideas for future direction, and the way that the Jamboard input that we got 2 years ago is being used to influence those future directions and ways that you can get involved. So we'll hear quickly from each group. We're going to start with Veronica Corella, who is our leader for the Healthy Alternatives Work Group. We'll then go to Cleo Braver for the Sustainable Ag Work Group. And then I'll fill in with a few remarks about the Research and Data Gaps Work Group. We did hear more from that group at the last conference, so I'll keep it short and sweet. But let's go ahead and we're going to start with Veronica and the Healthy Alternatives Work Group.
Great, thank you. I'm Veronica Corella. I'm Legislative Director for the Maryland Children's Environmental Health Coalition, and I co-facilitate the Healthier Alternatives Working Group along with Steve McDaniel. This year, the Healthier Alternatives Working Group completed several projects that were informed by the Jamboard suggestions from the 21-22 sessions. First, we launched a new social media campaign for managing mosquitoes without pesticides. This was in response to several Jamboard comments about educating the public about mosquito spraying and making our educational fact sheets more visible on social media. The resulting social media toolkit for managing mosquitoes without pesticides campaign gained much more visibility on Facebook, X, Instagram, and Nextdoor platforms. We also updated and created a social media campaign for one of our very popular booklets called Pesticides, Risks, Prevention, and Healthier Choices. We've created visual posts that link to individual fact sheets. This fall, we did a letter campaign targeting Maryland public school systems to remind school officials, operations folks, facilities, and health employees about the need to pay attention to what disinfectants are used in the school building around our children. We reminded them about one of our great resources, saferdisinfectants.org, that is available to everybody for identifying the safest alternatives, no matter what needs disinfecting. Next slide, please. Some of the Jamboard ideas you see here summarize the numerous ones with similar themes. We identified several projects that the work group will pursue over the next year. First, we're going to publish the Pesticides, Risk Prevention, and Healthier Choices social media toolkit and share this with organizations to help spread the word. Watch for an email in January with a link to the toolkit and a request that you share the campaign on your social media pages. We plan to develop several more social media campaigns, such as the meaning of that yellow turf flag, safer lawn and land care alternatives, gardening for pollinators, and linking to resources on MPEN's website and other websites like Montgomery County's, which has great resources for pesticide-free lawn and land care. In fact, this conference was a direct overflow from the suggestions about educating on soil health and organic regenerative agriculture, and we will continue to discuss more ways to carry these ideas forward in the coming year. Next slide, please. The Healthier Alternatives Work Group is for stakeholders from diverse backgrounds who are interested in educating the public. It's a great place to share your expertise to fuel ways to build public awareness. Please look for an email inviting you to join us at our next virtual meeting in the first quarter of 2024. Thank you so much.
Thank you, Veronica. Impressive work. Awesome. Thank you so much. Let's jump over now to Sustainable Agriculture Work Group and Cleo Braver.
Hello there. Good morning. I'm Cleo Braver. I'm the co-owner with my husband of Cottingham Farm, which is out here in Talbot County, where we grow organic produce and we and organic fed rotationally grazed animals. So I am the co-facilitator along with Errol Maddox of the Sustainable Ag Working Group. So this year, the working group worked on a few projects. One of them that we're currently finalizing is a social media toolkit for educating the public on regenerative ag practices and what the benefits are of regenerative farming to the environment and to health, both personal on a micro scale and on public health on a macro scale. And the point is really to help drive demand in the public sector for supporting farmers who are using these regenerative practices. And so we're going to be launching that campaign in the first quarter of 2024. One of the things that I was pleased to help work on this year was developing a landowner action plan. It's a document that lists 20 different ways that landowners, and we mean big and small, as well as those who are actually farming land in Maryland or who permit others to farm on their land, ways that they can improve the health of their land and contribute positively to the Chesapeake Bay watershed at the same time. So recommendations, are going to be along the lines of, no surprise, moving away from synthetics, both synthetic fertilizers and synthetic pesticides, to more organic things, putting buffers and cover crops, and even if it's not if it's ag land for farming, just like gardening, moving away from synthetics, moving away from lawns to native plants and shrubs. But even those who aren't landowners can be a part of this transition because we are all consumers of food. So you don't have to be a landowner to participate. And then one of our working group members, Steve Kraszewski, who you met this morning during Oren's talk, is working with us to develop a video series on organic regenerative farming practices. It's aimed at farmers who may be interested in transitioning from conventional to organic healthy soil practices, much as the Masons did a number of decades ago. He's already finished and hope we'll be able to view soon his first video on strip till corn. So thank you, Steve, for that work. We're excited to see it. Next, there we go. Our working group also evaluated the Jamboard ideas from 2021, and we're looking at ways to incorporate those ideas into our work in the coming year. The ones that you see here are the ones that we are likely going to be focusing on: sustainable land practices, making public and private landowners aware of practices that they can engage in, pesticide dangers and safer alternatives. We can grow our food and our fuel and our flowers and our plants without those synthetics. Pollinator protection is going to continue to be important to us, and building up healthy soil and, as a result, storing carbon. So those are going to continue to be important for us. We'll finish the landowner action plan and publish it this year and distribute it to landowners and farmers in Maryland. And Steve is going to continue to develop and create more of his great short video clips on organic regenerative farming practices. And lastly, we're going to be launching our organic regenerative social media campaign and be asking you again to spread the word about that. So watch for an email in January with a link to the toolkit and a request that you share that campaign with on your own social media pages. Again, this conference topic was in response to your interest in healthy soil and organic regenerative ag, and we're going to continue to work on that in 2024. So, this working group is for farmers, for gardeners, for land care professionals, for any stakeholder in our agricultural ecosystem, and that includes those of us who eat, because by eating food, we are taking an act that dictates what our food production practices look like. So it's a great place to share your expertise on farming or on gardening, and even legal issues for farmers. So look for an email inviting you to join us, for those of you who haven't already, at our next virtual meeting, which will take place, I believe, in the first quarter of 2024. So thank you.
Thank you, Cleo. Great work. I cannot wait to see that video. Really exciting. Okay, so let's just touch quickly here on the research and data gaps work group. Looking back, two principal efforts in the work group. The first is a report that we mentioned at the last session, communicating recent pesticide research findings to inform stakeholders. This is approximately a 10-year look back for studies that we think are particularly relevant. Maybe they are specific to Chesapeake Bay watershed. If not, we think they have the potential to be relevant and informative to the watershed's science community and policy community. So we looked back and we summarized in the end 25 studies that we thought were interesting and relevant and important. And we're finishing up that report, putting the last couple finishing touches on it. The other was that we did an update of work that you may remember if you've been with us for a while. We created a methodology for ranking our relative concern for different pesticide active ingredients. The idea was that we know there are hundreds, if not thousands, of active ingredients being used in the watershed. And what's most important? How do we focus in and prioritize on things that appear to be the most important? And this methodology used a range of data about use and potential impact and risk. And we have done an update of those matrices. So we'll be sending something out on that and making sure that you have access to the data and the conclusions that we've drawn there. So that's the 23 look back, yes. And generally speaking, we saw a lot in the Jamboard asking us to continue to follow the research on pesticides and the environment and people. And also there was a strong theme in 21 of interest with regard to PFAS in pesticide products. So we've followed that through here. I think the products that I just described, including our look back report and the updated matrices, are directly addressing the first bullet there. And we're bringing back some PFAS updates in this year's conference. So we'll learn more about that. One thing that we're hoping to do is, instead of waiting to do a 10-year look back on key research findings, we'd like to do that throughout the year so that at each conference, we can bring you some summaries of key studies and published research that we think is important, and bring it to you on a yearly basis instead of a long-range look back. So we're going to try that this coming year and make this update just a routine part of our annual conference, so we're up on the absolute latest research findings that we're aware of. I was looking forward to 2024 and the ways that you can get involved here. So along that specific point, if you see a research report, we prefer to use peer-reviewed, published journal reports if possible. There are some other things that are credible, some other sources. So if you see any of those that you think are really important and this community of people that comes together each year could benefit from, please send it to us. Of course, we'd like to have you or any other stakeholders or experts in this area that might help us with our research and data gaps work. And as I think has been mentioned, we're going to be recruiting for the work groups. We show here how you can get involved. We want to hear from you. We want your expertise. It's not a great big time commitment. We don't tend to meet very, very frequently, but there are a few meetings a year or so. And if you jump in and start helping with a specific project, well, that might involve some more time, but love to have you not only just attending, but helping us. And I think you can see, again, this point that I make, that this is really something that is special about this project. We don't just come together and talk every year. We also come back together and work and produce products that hopefully are making a difference. And we would love to hear from you and have your involvement. There will be an email that will go out and we'll have some questions for you and hopefully encourage you to work with us throughout the year so we can bring back the kind of work products that you heard about just now each year in our conference. All right. Well, thank you so much, Veronica and Cleo, for your leadership and for the great products that are coming out of those work groups. So we're going to switch now to the last portion of our program. And I'm going to introduce to you Dr. Nathan Donnelly, who's going to give us an update on what we're learning about the presence of PFAS in pesticide products. Dr. Nathan Donnelly is the Environmental Health Sciences Director for the Center for Biological Diversity's Environmental Health Program. They work on issues surrounding the increasing exposure of both people and wildlife to toxins. Before joining the Center for Biological Diversity, he worked as a scientific researcher in the Oregon Center for Research on Occupational and Environmental Toxicology. There, he was studying the links between exposure to environmental toxicants and cancer. Dr. Donnelly holds a doctoral degree in cell and developmental biology from Oregon Health and Sciences University. Thank you, Nathan, for joining us with this update today.
Well, thank you very much for the introduction. I'm super excited to be here today talking about some research that some colleagues of mine have put together over the last year or so. And I just want to mention some of the data we'll be presenting today on the slides are currently under peer review right now. So they're, should be considered preliminary data. Just want to be upfront about that. So next, I'm going to talk today about PFAS and pesticides. And before I jump into pesticides, I want to make sure we're all on the same page about PFAS because things have been changing in this field quite rapidly. And there's a lot of misconceptions out there about what PFAS are and are not. So PFAS is just an acronym for per and polyfluoroalkyl substances, which is a pretty meaningless term. It's incredibly broad, and it's been interpreted very differently by many different organizations around the world, really, and even been interpreted differently by the same organizations over time. So there's just a lot changing here in what is or is not considered a PFAS. I would say most people, when they think of PFAS, think of specific examples of PFAS like PFOA, PFOS. These are the most infamous examples. Long chain PFAS, incredibly persistent, incredibly harmful. PTFE, which is that nonstick coating in your frying pan and in that bottle of Scotchgard that your mom used to spray on the furniture to make it stain resistant. Gen X is what's called a shorter chain PFAS. It's supposed to be a replacement for PFOA, and it's turned out to be just as persistent, just as harmful. So these are kind of the dirty four, if you will. But the more we're learning about the persistence and the toxicity of some PFAS molecules, it's become very clear that we need to be looking at more than just the worst of the worst examples. So on the right here, you'll see a structure of PFOA, and the C's are carbon atoms and the F's are fluorines. And you'll notice that it's basically just a long chain of carbon fluorine bonds. This is called a long chain PFAS. It's got 7 or more fully fluorinated carbons in its chain. It's a pretty nasty molecule. There are also short chain PFAS, which has fewer than 7 carbons in the chain. And then a subset of short chain PFAS are ultra short chain PFAS, which have generally 1 to 3 fully fluorinated carbons in the chain. So I'll be talking a bit about chain length as I go along in this presentation. So just be aware that the PFOAs and the PFOSs, those are long chains, and we've got shorter chain things like Gen X, and then there's ultra short chains, which just 1 to 3 fully fluorinated carbons. So there's 2 really main PFAS definitions in use in the US right now. And this has been relatively recent. So a few years ago, the OECD, which is the Organization for Economic Cooperation and Development, it's an international standard setting agency that EPA and many regulatory agencies around the world often look to develop their own thresholds and definitions and so on and so forth. So it's a highly regarded organization. The OECD definition here, I'm going to refer to as the scientific definition. It was developed very transparently, published in peer review by a group of researchers from around the world. So I'm going to refer to this as a scientific definition. The OECD definition is basically any molecule that has at least 1 fully fluorinated carbon. And what that looks like is just a CF3 group on the side of the molecule, or in some cases, a CF2 group. This is in use now in some states who are opting to regulate PFAS above and beyond what the feds do. It's also used in some other countries around the world. On the other hand, we have what I'm calling the regulatory definition. So this is a definition developed by the EPA Chemicals Office, again, a few years ago. And it is any chemical that has 2 adjacent carbons with varying degrees of fluorination, at the very least, will meet the regulatory definition of a PFAS. This is used, again, in federal regulation. And while these definitions look kind of similar, they're actually very different. The OECD definition is very broad. It covers about 20,000 or so different molecules we know to be in existence. The regulatory definition encompasses about 7,000 or so. So it's a much more narrow definition. And we don't quite know the origins of this definition, the regulatory definition. And that to me is a major red flag. So moving forward in this talk, I'm going to be using the OECD definition of a PFAS, but just know that there may be some disagreement just based off of differing definitions that different agencies are using. So why all the fuss? Who cares if a molecule is fluorinated or not, right? Well, it turns out the carbon-fluorine bond it doesn't exist in nature. I mean, there are a few, of course, exceptions, but for the most part, any chemical out in the environment with a carbon-fluorine bond is generated by humans and released as pollution. And so that means that no biological processes have evolved to break down that bond. It's incredibly strong. Bacteria don't break it down, fungi don't break it down, algae don't break it down, sunlight doesn't break it down, heat, pressure, it's just there. It's not going anywhere. And that's really worrisome from a persistence point of view. So that's why PFAS have earned the moniker forever chemicals, because that highly fluorinated portion of the molecule is going to exist forever. We're talking your grandchildren's grandchildren will be encountering the same molecules in some cases that we are encountering today, which is really worrisome. I will say the very few PFAS that have been studied in any depth are, again, very persistent and really incredibly toxic at very low doses. So it's an issue that deserves our immediate attention. Next. So how did these nasty forever chemicals get into pesticides? It turns out there's a few categories here that they can be broadly lumped into. One is that some PFAS are intentionally added to pesticide products. The pesticide makers are putting them in there, and they want them in there for the chemical properties that they have or whatever. And then there are unintentional ways that PFAS are included into pesticides. Ways that the pesticide manufacturer did not necessarily intend on. So I'll give you a few examples of these moving forward. Next. So I'll start with active ingredients. This is, again, intentionally added PFAS. So active ingredients are the primary component in the pesticide. It's the chemical that is killing the pest that you are targeting. It's the main ingredient in the pesticide product. And so for this, what we did was we looked at all conventional active ingredients that are approved by the US EPA for use in the United States. So conventional ingredients are what you think of when you think of a pesticide. It's the stuff used in agriculture. It's the stuff used around people's homes to target things like insects and fungi and weeds and stuff like that. We're not looking at biopesticides, which are things like natural extracts or living organisms that are used often in organic agriculture. We're also not looking at antimicrobial pesticides, things like bleach or triclosan, which are used, to generally sterilize indoor environments. So we're just looking at conventional active ingredients approved in the US. There's about 500 or so. And I'll direct your attention to the green bars here. So this is looking at all the 500 active ingredients approved in the US right now. About a quarter are organofluorines. Organofluorines are just molecules that contain at least one carbon-fluorine bond. So about a quarter of active ingredients approved in the US have at least one carbon-fluorine bond, which is pretty high. And then if we look over at the second column there, the green bar again, about 15% of active ingredients approved in the US, conventional active ingredients, are PFAS, meet the scientific definition of a PFAS, which that's like 1 in 7. That's really high, much higher than we were expecting to see. But the trends are looking much, much worse. So the blue bars now are just looking at all conventional active ingredients approved just in the last decade. So we're looking at the most recent approvals. Over 60% are organofluorines. It is now the norm to fluorinate active ingredients. That is the trend in the industry. That is what they are moving to. And about 1 in 3, almost a third, are PFAS of recently approved active ingredients. So, we're seeing this, this seems to be desired by chemical manufacturers now to fluorinate, and in some cases highly fluorinate their active ingredients, which from a persistence point of view is pretty darn worrisome. Next. So what does that look like? I would say most, almost all of the PFAS active ingredients just have a CF3 group, just one fully fluorinated carbon attached to the molecule somewhere. So it meets the scientific definition of a PFAS, but most of them do not meet EPA's PFAS definition. Some of them do though. There's a handful, maybe 2 or 3, that actually meet EPA's definition, and they're pretty gnarly looking. This is broflanilide up here, is approved just a few years ago. Highly fluorinated, highly persistent. We're talking about soil and water half-lives of 5 to 6 years. So a half-life is just how long it takes for half of what you put into the environment to break down. So we're looking at, if you spray this in the environment, it takes 5 or 6 years just for half of what you spray out there to begin to break down. And that's just the time it takes for one of these bonds to break. So, then the clock starts over. So what we're looking at is likely over decades, this thing will be slowly chewed up into the environment until we're left with, probably that top fluorine structure, maybe even that ring structure with the other fluorine attached to it. And that's not going anywhere. That is a terminal degradate. We're talking about half-lives on par with DDT. Again, highly persistent, very, very worrisome that we're seeing these structures represented in active ingredients right now. Next. So I'm going to switch gears now, looking at inerts. So we're still looking at intentional addition of PFAS. Inerts are every other ingredient in the pesticide product other than the active. So things like dyes, fragrances, surfactants, emulsifiers, propellants, things like that. Anything else other than the active ingredient is, quote unquote inert, as an inert ingredient. And they often account for the bulk of the product. Some products are like 99% inerts, other ingredients. So they can be really a major source of the chemical addition into a pesticide. Inerts are shrouded in secrecy. There is little to no transparency about what products these inerts are in. They're not required to be put on the label. The public has no clue what inerts are in pesticide products. Healthcare workers have no clue what's in the inerts, what inerts are in pesticide products. So it's from a public right to know point of view, very worrisome. We know what active ingredients are in products, but we have no clue about any of the other ingredients. That's considered trade secret, confidential business information. But we have been able to glean a few things from public records requests via FOIA. It looks like there are about 11 fluorinated organofluorine inerts registered with the EPA, and 8 of those meet the scientific definition of a PFAS. So much lower numbers than we were expecting to see, which is a good thing. However, one of these is, and you can go to the next slide here, PTFE, which is the better known as the brand name Teflon. This is an approved inert ingredient in the United States that is present in some pesticides. That non-stick coating in your pan is present in micronized form in some pesticide products that are approved in the US. Very worrisome. PTFE is highly, highly persistent, and spraying it out in the environment in a broadcast fashion is incredibly worrisome. So I'm hoping that this will get canceled very soon. So we've got 8 PFAS inert ingredients approved in the United States. And we actually more recently identified how many products these inert ingredients are present in. This is, again, information through public records requests. And the good news is it doesn't look like they're in all that many products. Right now, the 8 PFAS inert ingredients approved in the US are present in a total of 55 pesticide products. And we even looked at Canada. They have about 7 PFAS inert ingredients approved in that country that are present in just 41 products. So given that we have 20,000 or so pesticide products approved in the US, these numbers are very small, and that's really good. However, I want to highlight the lack of transparency here with inert ingredients because we don't know what these products are. They could be products that are very lowly used, or they could be products that are used across millions of acres across the country. So I want to caution that this isn't necessarily good news, although the low number of products is great to see. These could be widely used products. We just don't know. EPA won't give us any information on what products contain these inert ingredients because, again, it's considered confidential business information. So all we have is the number of products these inerts are in, and that's it. So I'm, again, hoping moving forward, we can get just a little bit more transparency here to the extent the EPA is able to try and figure out, okay, what products are these in? Are these very widely used? Give us something here because, again, there's a lot of uncertainty here with inerts. So we can go to the next slide. Now we're looking at unintentional addition of PFAS into pesticide products. And it's looking like container leaching is going to be a major pathway for that to happen. So a little history here. A few years ago, peer public employees for environmental responsibility found levels of long and short chain PFAS like PFOA and GenX in parts per trillion, parts per billion concentrations in multiple mosquito insecticide products that were being used in Massachusetts, and the state of Massachusetts confirmed that. EPA did a little rooting around and decided that this contamination was likely coming from the containers that the pesticides are housed in. So it turns out that pesticides are pretty caustic, and they can actually over time start to break down high density polyethylene that they're being housed in. And one way that manufacturers have gotten around this is to fluorinate those molded plastic containers. So what you do is you put them in a chamber, you hit them with fluorine gas at real high temperatures and pressures, and it changes the carbon hydrogen bonds in the polyethylene basically to carbon fluorine bonds on the edges of those containers. So you're basically putting in a PTFE coating on those containers by fluorinating it. The problem is, it works to make it so that these pesticides aren't chewing up that plastic over time, but it produces PFAS. And this is now really a well-established pathway that PFAS can get into contents that are housed in these fluorinated plastic containers just by leaching from the edges of those containers. If you've got a pesticide product and you put it on your truck and you ship it across the country, it's going to be jostling around. As you drive through Denver, it's going to be exposed to really high pressures. As you drive through Phoenix, it's going to be exposed to high temperatures. All this is going to promote leaching of those PFAS that are adhered to the sides of the container into the actual pesticide liquid itself. And that's worrisome, right? Parts per trillion and parts per billion concentrations may sound very low, but when we're talking about PFOA, that is actually very high. Some of these long chain PFAS really need to be thought of along the lines that we think of dioxins. Very harmful at very, very low doses. So parts per trillion, parts per billion concentrations is of human health relevance there. And we know that fluorination of rigid plastic is not just happening for pesticides, this is happening for food products too, which is really quite worrisome. Next. So now, a few years have passed, multiple groups, including my own, have found parts per trillion and parts per billion concentrations of long chain PFAS in pesticide products using a third-party certified lab. So this is Eurofins Scientific, which is one of the labs that kind of helped write the book on PFAS testing back in the day. We're finding levels of long chain PFAS that are not approved active or ingredients, so they shouldn't be in pesticides. We're still finding them in some pesticide products, and it's really worrisome. It's looking like some of this is coming from container leaching. Recently, EPA has done its own testing and failed to reproduce some of the findings that has been identified through Eurofins. So the pesticide office has been doing some of their own testing of pesticide products, and they're not finding PFAS that other labs are finding. So there's lack of reproducibility here, which is worrisome for me. And so it really highlights the need for us to figure this out and standardize reliable testing, because we need to be able to reproduce results, and that's not happening right now. Given the persistent nature of especially long and short chain PFAS, we need to figure this out quickly. And I'm hoping that our regulatory agencies are taking this as seriously as I really think it should be. So container leaching is clearly one major pathway that long and short chain PFAS can get introduced into pesticide products. But it's looking like there are other unknown sources. And the reason I think that is because the testing that has been done that has been finding some of these long and short chain PFAS in pesticides are also finding long and short chain PFAS that are not known to be produced via container leaching. So container fluorination produces carboxylic acid PFAS. but we're finding some sulfonic acid PFAS in some of the testing that's been done. Those are absolutely not coming from container leaching. So there appears to be other, as of yet, unknown sources. Possibilities include, impurities from other ingredients that are being put into these products, or just simple contamination of the water that is being used to dilute these pesticides in the bottle. We're not quite sure where these PFAS are coming from, but it's looking like container leaching is not going to be the only pathway for these really nasty ones, the long and short chain PFAS, to begin into pesticide products. So again, a lot more work needs to be done here. We need to figure out how these compounds are getting into pesticides, because we need to get rid of it immediately. Next. So I talked with you earlier about active ingredients, PFAS active ingredients. I want to go back and touch on that again, because a lot of people might be saying, who cares if a active ingredient is a PFAS, right? EPA has done their risk assessment. They've assessed risk. They've made sure that that approval has been done in alignment with federal law, no unreasonable adverse effects to people or the environment. Who cares if it's a PFAS or not, right? EPA has done its due diligence. The problem is the risk assessment process is not really designed with persistent chemicals in mind. I would say most pesticides, certainly the majority, are not highly persistent. And the risk assessment process is really designed to look at the parent pesticide molecule and a couple of the degradates that are formed early on, and not much else. So EPA guidelines right now require that if you are a pesticide maker, you need to submit studies to the EPA to analyze degradation of your pesticide, which is good. The problem is those experiments last at most 4 months. So we're looking at degradation over the course of months. That's fine for some pesticides, but I would argue for fluorinated structures in particular, chemicals that have, half-lives, or at least they're degradates that have half-lives likely in the decades or centuries. This gives an incomplete picture about what's happening. We can't really tell much 4 months down the line about what is ultimately happening to these guys. What are the terminal degradates, the fluorinated terminal degradates that are going to be around forever? We simply don't know that with a lot of these pesticides. And sometimes even highly persistent degradates that are known to be formed are ignored. And I'll give you a few examples. One is sulfoxaflor, which is a PFAS AI active ingredient. In EPA's risk assessment, they acknowledge that there is a highly persistent fluorinated degradate that is, coming from the sulfoxaflor parent, that is, from the parent metabolism into this highly fluorinated, highly persistent degradate that was likely to accumulate in the environment over time. EPA looked at a few acute toxicity studies to aquatic invertebrates and decided that it was not as toxic to those organisms as the parent sulfoxaflor molecule. So they said, it's not a risk. We're not going to assess it further. Same thing with bicyclopyrone. Again, you've got multiple highly fluorinated, highly persistent degradates being formed. EPA didn't even look at studies in this case. They just did a structure-activity relationship with these degradates and said, well, it doesn't have the active structure that we know is responsible for bicyclopyrone toxicity. Therefore, we're not going to assess risk from it. So, kind of worrisome. I mean, there's a ton of uncertainty there. Just because it's not toxic on an acute basis to aquatic invertebrates really tells you absolutely nothing about its toxicity to other taxa, to people. And again, if these are highly persistent, we're talking about half-lives in the decades or centuries. If you're wrong, if this is really toxic, it turns out that we find that out 10 years later, there's nothing that can be done. You can't reverse this. So the uncertainty involved in what degradates are formed, how toxic they are, really necessitates that we need to be monitoring for these guys, and that is just not happening. So if you look at this little table over there, we have about 66 conventional PFAS active ingredients approved in the US. Only 13 of those have been monitored in water by USGS. USGS did a great extensive study about 10 years ago, actually a series of studies where they looked at about 230 different pesticides and their degradates across 500 streams in 5 different regions in the US. Really extensive stuff. But they only monitored 13 for 13 of these PFAS active ingredients. That's about 20%. And of those 13, they found 12. While a couple of these were just found in 1 or 2 streams, most of them, are some of the most widely detected pesticide water contaminants in the country. Things like bifenthrin and isoxafludol and fipronil, trifloxystrobin, sialothrin, contaminants that are showing up in waterways across the country. And we just don't have a good sense of what degradates are being formed from these. And we're not, the federal government is not really monitoring most of these out in the environment at all, at least in water, which is very worrisome. I'll also say that it's looking like, just from the structures of some of these active ingredients, one likely degradate from most of them, one likely terminal degradate is trifluoroacetic acid, or TFA. TFA is a widespread pollutant across the world, really, showing up in water, showing up in food samples, showing up in human serum, urine, drinking water. It's really kind of everywhere, and it's just now being appreciated as being a serious part of the total organic fluorine load in the environment. And it just so happens to be a degradate of many PFAS active ingredients. And that degradation could be extensive. So the German Environmental Ministry a few years back, which is Germany's EPA, looked at TFA in the country, and they found that they had 28 PFAS active ingredients approved in the US. So there's only 28 approved in Germany. Of those 28, Germany found that up to 500 tons of TFA could be produced just from the degradation of those 28 active ingredients in the country. Given that we have 66 PFAS active ingredients used in the US, and we probably use many orders of magnitude more pesticides than Germany does, I think pesticide degradation is a major source of TFA in the environment. There are certainly other sources as well, but this is worrisome. And since you have dozens of PFAS active ingredients likely ultimately degrading into TFA, we need to be assessing the cumulative impact of that. The problem is there's no cumulative impact assessed. EPA is taking a chemical by chemical approach. And when they're all degrading into the same thing, that can be worrisome. There could be risks there that you don't understand if you're just doing a chemical by chemical approach. So I'm hoping that with some of these fluorinated active ingredients, EPA can undertake a more extensive evaluation, because it's certainly warranted. And again, like I said, any release of these highly persistent degradates into the environment, again, trifluoroacetic acid, TFA, has a half-life of 200 years. We are talking about something that is just not going to break down. And it's getting into our bodies, it's getting into drinking water and food. It's being found in wildlife in remote areas. Again, very worrisome. Next. So I'll just leave you with this. This is a quote from a colleague of mine, Dr. Kyla Bennett. So it's looking like unintentional contamination, and right now we're assuming to a lesser extent inerts are the likeliest sources of long chain PFAS, like PFOA, PFOS, and then in the case of inerts, PTFE again is an approved inert that's present in pesticide products that are used in the US. Active ingredients are clearly the major source of what we're calling ultra short chain PFAS. So this is 1 to 3 fully fluorinated carbons. And they're present in much higher concentrations. When we're looking at parts per billion, parts per trillion concentrations of some of these longer chain PFAS and pesticide products, active ingredients are present in parts per 100 concentration in these pesticide products. So much more concentrated. We know from estimates from USGS that we use at least 23 to 35 million pounds of these PFAS active ingredients each year, every year, year after year in the US. And that's just from 2018 data. There are many more that have been approved since then. So I think it's likely we're looking at between at least 30 to 40 million pounds used each year of PFAS actives, since many of them have been approved more recently. And again, only 20% of them have been actively monitored by the US government and water. And the ones that are detected are some of the most widespread pesticide water contaminants in the country. Again, whatever we put it out into the environment today, even if we think it's benign today, tomorrow that could be a very different picture. And there's no reclaiming what we're putting out in the environment. There's no reversing it. It's done, which makes this something worthy of really figuring it out and getting right. And I'll just leave you with this. If the 60s and 70s were the age of the organochlorine, things like DDT, aldrin, chloridane, we're living in the age of the organofluorine right now. And there are going to be legacy impacts, but what those impacts are is just not understood right now. And that's scary. I mean, DDT is still one of the most widespread pesticide contaminants in meat and dairy in the country. And we haven't used it in 50 years. So, we're talking about legacy impacts here that we can't even begin to comprehend right now, even with, compared to most chemicals, a lot of the study that's been done with some of these pesticide active ingredients. There's just a lot we don't know. And that uncertainty really worries me. So that's all for me, and happy to take any questions from anyone if you have any.
Thank you very much, Nathan. You certainly make a compelling case for concern. And it's clear that this topic is still nascent in the science, and the complexity of the risk assessment that's needed for this. Really a strong case on those points. So we are coming close to our time here. Just a question or two. We understand that the company that has been the primary producer of the fluorinated containers has been ordered to stop that process. And the question is, do we know that there are other providers? Are there other containers that perform well enough that there's an easy replacement there? What is the replacement picture?
Yeah, there are replacements. So for those of you who don't know, EPA a couple weeks ago just took action to basically order the company that's producing all these fluorinated containers to stop producing PFAS. And since you can't really fluorinate containers without producing PFAS, it's basically an action to stop the production of fluorinated containers. And that's supposed to take effect February of next year. So it's coming up. So that's a great action. I mean, a very strong, bold action from EPA there that could really cut down on a source of PFAS in pesticides, as well as food and other products as well. I will say that, the company has challenged that action, and the Fifth Circuit Court of Appeals just stayed that decision. So that won't go into effect until the court comes to a conclusion on the legality of EPA's action here. And since it's one of the first major actions under the newly revised Toxic Substances Control Act, I wouldn't be surprised if it made it all the way to the Supreme Court, to be honest. So read into that what you will. But suffice it to say, there's a lot of uncertainty now on whether that action will take into effect or not. If it does, I will say that it will cut down on a major source of contamination of PFAS in pesticides, which is great, particularly long chain PFAS. And there are other options. I'm not, that familiar with them. I know one option is to sort of do what's called an in-mold fluorination. So instead of taking your plastic containers and then blasting them with fluorine gas, you kind of do the process while the plastic is being molded. And that, at least as far as a few studies have suggested, does not produce PFAS, at least to the extent that post-mold fluorination does. So there are other options. There are also non-fluorinated ways of making barriers in these plastic containers. So I really hope EPA's action takes effect, but we won't know for some time until that will take place or not.
Okay, well, that sounds like a positive development. And maybe that's a positive note that we can end on here. We could talk with you for hours, probably. Unfortunately, we're up on time. Once again, there's a few questions in chat and Q&A that you might respond to. Some interesting questions about biological processes for breaking down and whether there's any potential for biological remediation or mitigation for PFAS. So if you can visit those, we'd appreciate that.
Yeah, absolutely.
Okay. Yes. Well, thank you so much for that, Nate. And maybe we'll be back again next year with another update.
Right.
Okay, great. Yes. Bonnie.
So much. I have learned so much from this year's presentations, and I hope you all have too. I also want to let you know that we're going to be sending out that email with a survey link where you can give us feedback on the conference presentations. And we're also going to be sending you links so that you'll be able to access all of these presentations on video, as well as the PowerPoint presentations, the chat transcripts, and the Q&A transcripts. So we'll be able to get that out to you probably late next week, and you'll have all that great data as well as contact information for one another. So we just really want to, again, thank you for joining us today. And we'll be sending you that invitation about the project working groups. We hope you'll become a more involved part of our community. And I want to thank Greg Allen, our most terrific moderator, and also the wonderful presenters, Orrin Valcourt, Steve Kraszewski, Catherine Dalton, Nathan Donnelly, Veronica Corella, and Cleo Braver, as well as our sponsors, funders, and the team behind the scenes, Emily Ranson, Sean Lynch, Doug Miller, and Ruth Berlin. So I know we promised to get you out, and I'm going to end that with one more slide that you guys can leave on, which are our 3 websites that Maryland Pesticide Education Network has produced. And these are great places for you to take next steps, whether that's data that you want to research, or studies you want to access, or ways to become more organically motivated. Next slide for that. And we thank you so much for being part of us and part of this project. And we'd like to wish you a great holiday and new year. Thank you so much, everybody.
And also thanks to Bonnie Raindrop. So happy holidays, everyone. We'll see you in 2024.